Good morning everyone. Now in the third lecture we are going to start about the different components of the electron microscope. So before that, where we finished last class, we want to discuss little bit and continue that discussion, because in class 1 I already discussed about the resolving power of any lens or any microscope or other optical instrument. So we should discuss about the about the resolving power of any microscope. So here last class I already discussed about that. Okay, so relativistic effect will be there, and because of this accelerating stack, that electron is moving through the accelerating stack after it is coming out from the electron source, and it is increasing its speed, and that speed we already discussed that speed will be almost 70% of the light speed. And so that is the reason we always have to consider that relativistic effect, and the final equation we can get it, that is coming that lambda equals to h by 2 m0 ev, because mass will be modified, and into 1 plus ev by 2 m0 c squared, where c is the velocity, the speed of the light. So if we just take this formula, that okay, so there will be wavelength of electron, it is going to vary, and it is depending on the voltage of the microscope. And voltage means, you always remember that most of the time in electron microscope there are 3 different types of voltage we are talking about, that is 120 kilovolt microscope, then 200 kilovolt microscope, and 300 kilovolt microscope. This is called the high tension. When you will switch on, this is called the accelerating voltage, and this is basically helping us to accelerate the electron. And then definitely this voltage is playing a very vital role. If we remember last class also I discussed that, okay, so that means lambda is here inversely proportional with the root square of the voltage. So in this regard, that okay, at whatever we are calculating that 100 kilovolt or 200 kilovolt, last class I mentioned that okay, overall roughly that wavelength it will be 3.88 picometer, or you can say that 200 kilovolt, your wavelength it is coming 2.74 picometer, and then 300 kb it is coming 2.24 picometer. Now what is the actual velocity, actual wavelength we should consider when we are considering the relativistic effect? So here is a table where you can see it, that for 100 kilovolt, as I mentioned that it is in nanometer scale, so it will be 3.88 or 3.86 picometer, and this is 2.74 picometer that I mentioned last. So you can see there is a change in because of this relativistic effect. Here you can see this converted to 3.70 in nanometer, or you can say 3.7 picometer here, or this one will be 2.51 picometer, what was previously 2.7. And now here you can see for the 300 kV there is a drastic change. So previously it was 2.24 picometer, now it is coming 1.97 picometer. So you can see that okay, if wavelength is changing, is changing, and based on the voltage. So if we have a higher voltage microscope, this wavelength will drastically change. Now how this wavelength is helping us? So one thing is that when the wavelength is shorter and shorter, it will really help to penetrate through our sample, as well as it is going to affect our resolution. How it is going to affect our resolution? Let's take a look. Here the Fraunhofer diffraction pattern, we all know that okay, if there is a pinhole, the light will go through this pinhole and it will form an image somewhere here. So here if we just consider that we have a pinhole, if we just consider that okay, we have a pinhole and we are expecting that it should form one dot. So if we are trying to image this particular dot which is here, and we are expecting that we can image this dot as it is using any lens system. Even if that lens has no aberration and no other defect, if we expect that we'll get a perfect dot here, which is completely impossible, because in this particular case there is some blurring effect will be always there. So if we just consider eye is the best detector, and then any other microscope or camera, whatever we want to use it, there will be some kind of defect will be there, and that is the reason if we want to image a dot, we cannot image a dot anymore. So it will be some kind of halos will be there outside of this dot. So that is called the point spread function. So if we have a object and what is the relationship with object and what is the relationship between that object and an image? So we always say that image equals to, it will be object, then it will convoluted with point spread function, and point spread function, it is a blurring effect and it is blur the actual object. So we cannot get the perfect object. So that is called the point spread function. So if we want to image a dot, most of the cases we cannot get a dot easily. It will be some kind of halos, it will be there surroundings to that dot. So if we do a cross section of this dot, what we will get it? It will be some kind of Gaussian curve here. And if we want to image this dot, means we are just recording this amplitude. So we'll come back to those points and we'll discuss more. But here what we can see, if we want to image a dot, we'll get always a disk kind of thing, that is called array disk. So this array disk, if there is a two dots are closely packed and they are very close to each other, and both the cases it is forming this circular disk or array disk. So this disk can attached like this one. If they are attached like that, they are very close to each other but they are completely separated, that is the time we can tell that this is the maximum resolving power of that instrument, because these two dots are making a disk but they are separated from each other. So when these array disks are separated from each other, then that is the resolving power of a microscope. That criteria, it is coming from Rayleigh's criteria, where he proposed that one, that this is the minimum distance is required to resolve 2 points. Here I want to give one more example. So if we just consider that one person at night walking from far away and he is holding a two torch in his two hand, and both the torches are holding very close to each other, but that person is very far away from you and that person is approaching to you. When he is very far away and if it is a completely dark, you cannot distinguish two torch. It will appear like it is something, a big light is coming. But if that person is slowly approached to you, after a certain time you can see there is a two torch and two large discs are separated. So that is the point you can tell this is the resolving power. So where you can see there is a two searchlight or torchlight are separated. So here also we are discussing about the same thing in a very high magnification. So if we go back to our PPT, we can see that in this particular case this is the Fraunhofer diffraction pattern, where you can see that light rays is going, if even there is a pinhole or this aperture size, it is taking this kind of blurred image, and if we just try to project it in a two dimensional way, we can project it like this. So if we have a disk, so sometime what's happened, if we go to the next slide, we can see it. So here this is called the point spread function. We have a very bright spot here, and we have a very, this is a very bright spot here. This is my object, and this is the blurring effect. This blurring effect is convoluted, and it is giving us an image like that. So any image, it will appear like that. Center part, it will be very bright, and then there will be couple of front band, it will be there. So this is called the first maxima, second maxima. Here this is the first minima, second minima, third minima, like that. Now here, okay, so here we want to discuss little bit more about how we are getting those kind of curvature here. So if we just consider that we have a pinhole somewhere here, or there is a opening and there is a light source, and light is coming from a different position. So we have a source here, and now light rays are coming from this particular source, and there is a straight light, and this is the image plane where we can see that these rays are going and forming an image here. On the other hand, those rays will also come and merge somewhere here. Other possibilities is this will go somewhere here, this will go somewhere here, and this will form. So now the similar thing is possible in this side also. Now if we just consider, I am not drawing that part here. If you just go like that, okay, so you can see the most of the rays even coming and merging here, which is giving us the first peak here. Then there is a gap, there is a minima, and then again it is coming one particular peak. So same thing is possible here. If it is coming somewhere here, these rays will merge and give us a peak here. So this is the first minima and then first maxima, and it will go like that. So how many times it will oscillate, it is depending on what is the intensity we are getting here after the diffraction. So if we just try to draw this one, what we will get it? We will get this kind of curvature. Okay, so this is for one source or one light. If there is a two lights are coming together side by side, you can say that okay, there is a one light and another light source is very close to each other. So there are two possibilities are there. So let's take a look what kind of possibilities do we have. So if we go here, okay, so as I mentioned that based on that previous diagram, what will get it, if there is a 2 point source are very close to each other, it will give us a curve like that. It will merge, it will come, and then if they are very well separated like that, and then the second one it will come like this. So they are both are separated. So there is a two array discs are formed, circular array discs, and they are completely separated. So our definition wise resolving power will be that when these two particles are very close to each other, even there is a source of light, these two sources will be connected with each other. So if we come here, so if these 2 points are like this, and then there may be a 3 possibilities, it will be like that. Okay, so one peak it will come like that, other one it will, okay, so there is a one array disc is here, other one is here. The other possibilities is this, so there is a peak, so there is a, they are merging. So you can see in this particular case these two array discs are overlapped with each other more. So if we just consider this area and this area, it is more because these two peaks you can see they are much more overlapped with each other. So now the last possibilities is this, that we have a peak here and they are, sorry, So we have a peak here, and the second one is also. So in this particular case you can see that both the rings are completely overlapped with each other. So in this one we so here you can see that both the cases they are overlapped with each other so much we cannot distinguish these two circle anymore here also. But in this case there is some kind of separation, but that is not enough to resolve. There is a two discs are there, but in this particular case we can see the discs are completely separated. So if we just consider this center to this center of this disc, this distance it will be so something like 1.22 lambda by d. D is the diameter of the aperture. So this will be the angle theta. We can take that, okay, it will be a some constant 1.22, and then there will be a lambda, and then this is the diameter of the aperture. And now here you can see this resolving power is completely depending on the lambda. If lambda is very big, resolving power be not that great, but if lambda is smaller and smaller, it will really help us to reach a very high resolution structure. So this is the diagram. You can see that Rayleigh's equation, this is completely separated. Here there is a this diameter is still you can see these 2 points are close to each other but not overlap completely. Here 2 point is completely separated and there is a huge distance. So we cannot consider this and consider this and say that this is the resolving power of the microscope. Here this is the situation which one we can connect with the Rayleigh's criteria to define the resolving power of any optical microscope or any microscope. Here we can say this is not resolved at all because these two array discs are completely overlap with each other. So here this is the formula from where we can calculate the resolving power of that optical instrument or any microscope, and this is we have to remember. And here, so that is the reason we discuss so much about the lambda, that when that wavelength is shorter we can get a much better resolution or much better resolving power of that microscope. So it doesn't matter, whenever we are using the light microscope we are using a wavelength in between 450 nanometer to 750 nanometer. So here the limiting power is the wavelength of that light. So even whatever microscope will try to make where the main source is light, we cannot reach a atomic resolution or near atomic resolution. If we really want to achieve a near atomic resolution, we definitely have to decrease the wavelength, which will help us to resolve a very high resolution structure. And that is the reason electron microscope is any kind of electron microscope has we are using a shorter wavelength, and that is has a penetration power and also very high resolving power, which is helping us to achieve a very high resolution when we are imaging of any biological samples. Now we are talking about electron microscope and how to use it. It will be better to know little bit about the history of electron microscope, what is the history and how we can get reach to this particular point when we can use the electron microscope and we can resolve a very high resolution structure. So we will go through that like what I did for light microscopes also. Here, so Ernest Ruska is the first person. So here this is the Ernest Ruska's first electron microscope. He discovered the first electron microscope in 1933. And that 1933 that magnification was 12,000 magnification, and this is the first microscope which is made here, and this is the design. And any microscope, we are still now using the similar kind of architecture where we have the electron source at the top and then different types of lenses that I'll show you in the later slide, and then there is a viewing chamber and there are multiple knobs are there, and this is the binocular. From through that you can image any sample. So this really helps when after that de Broglie's discovery that electron has a wave like property also, and that really helped to encourage the scientists that Ernest Ruska to use that electron to make an electron microscope. Now in the next stage you can see Manfred Vaughan is the another person who invented that scanning tunneling electron microscope in 1937, and the later stage 1940 Ernest Ruska developed his own scanning electron microscope. So first they developed transmission electron microscope, and the later stage they developed the scanning electron microscope, and that is the two microscope, two major component of this electron microscopy field. One is transmission electron microscope and another one is the scanning electron microscope. So now here I am showing you some different types of microscopes over the period, that starting from 1960, that is Siemens microscope is here, and then same company in 1960 you have another type of microscope here. Zeiss is also have an electron microscope in 1964, and it has a different capability. Initially it is a 100 kilovolt microscope and magnification is only 12,000x magnification, and then gradually it improved to 40,000x magnification, and nowadays whatever microscope we are using, it is a starting, as I mentioned previously, that it is starting from 120 kV to 300 kilovolt, as well as the magnification nowadays even you can go 600K magnification to image any material science sample or biological science sample. And this column mostly here you can see this is the viewing chamber. Overall architecture, as I mentioned, it is same. The high tension or accelerating stack is here, and electron gun will be at the top, where the electron gun means that is the main electron source and electron is coming through that, and here the multiple lens will be here, and this is the viewing chamber. And for all the cases you can see the overall architecture is same. So those are the internal part of those microscope. We are going to discuss little bit more about the electromagnetic lens. So this is the electromagnetic lens, this is the camera setup, this is the holder insertion, and most of the things are taken from Ruska's microscope. Those are the different component, and all these details are available online in from this source. So there are those articles are mainly written in German language. So here I tried to modify those things and showing it here. So now this is the overall architecture and drawing of this microscope. Now one important question it is coming that okay, so when in India we started developing our own electron microscope. I will come back to that point also. Here you can see some kind of modern electron microscope. Here I am showing you that JEM 1.25 megavolt high voltage electron microscope, where this is the microscope column and this is the part, the high tension tank is attached with this microscope. And there are few more microscope from Zeiss, Hitachi, as well as the Geol and then FEI. So there are different types of microscope and it evolved gradually in last 90 years. Now it is coming that okay, in chemistry in 2017 Cryo-electron microscopy got the Nobel Prize, and so that three scientists, the famous scientists, that Professor Richard Henderson, Professor Jacques Debouche, and Professor Joachim Frank, these three got the Nobel Prize in 2017, where we used the cryo-electron microscope for the development of this cryo-electron microscope. So what is this cryo-electron microscope? Cryo-electron microscope, it is a modified version of the transmission electron microscope, where the similar kind of transmission electron microscope we are using at a cryogenic condition. So we are preserving our sample at cryo temperature, and then we are imaging, and it was not an easy task, because in the microscope, electron microscope, everything will be under vacuum, and you have to maintain the temperature inside the microscope at liquid nitrogen temperature. So these three famous scientists really helped to develop this field, and then now last 10 to 12 years there are many biological macromolecules structure was resolved using this technique. So previously, whenever we are talking about that, for the drug discovery we need the structure, for vaccine design we need structure. Even if you think about during the COVID time, so many medicine vaccines evolved, so came into the market so fast, and if you look at the literature you will get it, there are many structure resolved with the drug, with the vaccine, using the cryo-electron microscope. So previously there is only two techniques is there, one is X-ray crystallography and another one is the NMR. But now the cryo-EM is playing a very vital role. You can keep your sample in the physiological condition, you do not need a very high salt concentration or very other artificial condition. Sample will stay at physiological condition, and a very small amount sample is required, and you can resolve the structure of any biological macromolecules. So here there is no size limitation, where in X-ray crystallography or NMR there is a size is a big problem. So here using cryo-electron microscope you can resolve the entire structure of a cell. So that is the good point, and that is the reason biologists are very much interested to use the transmission electron microscope and cryo-electron microscope to resolve high resolution structure, which we can use it for drug discovery purposes. Okay, as I mentioned that, okay, so Ernest Ruska first discovered that electron microscope, and then we are all using, but when it is started in India, if we look at in 1946, even before our independence, during 1946 to 1948, Professor Anand Dasgupta, he was a professor at Calcutta University as well as professor at Saha Institute of Nuclear Physics. He invented a horizontal microscope. So previously I showed you the perpendicular microscope, here you can see a horizontal microscope, and they invented that one, and that is the Asia's first horizontal transmission electron microscope. And the next question it is coming, who developed that or initiated that cryo-electron microscopy in India? So here there are couple of papers published from this group, where they use this electron microscope and image different microorganisms. And here the Professor Professor Amarnath Ghosh from National Institute of Cholera and Enteric Disease Electron Microscopy Division, he started that cryo-electron microscopy in 2001-2002 in India. Okay, so now we are coming to the main topic, that how we can use electron microscope for our study. So I finished the history part. Now we have to start and discuss about how electron is interacting with our sample, especially biological sample, and how we are imaging, what is the source of the electron. Those parts I will talk about in next class, and thank you all for your attention.